It's a pleasure to welcome our visitor today, Dr. Stacey Glasgow from Baylor College of Medicine in Houston. Dr. Glasgow is a member of the research faculty member in the Department of Neuroscience, a department that I know very well. And she's also a member of the Center for Cell and Gene Therapy as well, directed by Ben Deneen at Baylor College of Medicine. Dr. Glasgow did her undergraduate work at Trinity University and then went on to her Ph.D. at University of Texas Southwestern. And from there, she came to Houston, to Baylor College of Medicine, to do her first of a couple postdocs, initially in genetics with Dr. Lee, and then moved over to the Cell and Gene Therapy Center on Ben Deneen's group to do a second postdoctoral fellowship there. And from there has gone on to become a research assistant professor at the center, which is a really exciting center at the Baylor College of Medicine. So she's now a research assistant professor. She also holds an NIH K01 Mentored Scientist Award that she got about a year or two ago, I guess, approximately. And so she's obviously doing very well. Dr. Glasgow has studied white matter injury, and in particular the role of nuclear factor 1A and its role in inhibiting repair in white matter in the central nervous system, and also looking at expression profiles and astrocyte precursors within the spinal cord early on. She's gone on to really elucidate in great detail the diversification of glial cell lineages and looking at the transcriptome and interaction of signaling pathways and the role in proliferation and also in tumorigenesis in particular. I think we're going to hear some of that today. She recently has made contributions in lineage specifications, again, of NF1A in the developing CNS and also in glioblastoma or in gliomas, and looked at the role of modulation on transcriptional networks and the overall chromatin landscape and remodeling of chromatin. So this work, I think, is opening up new vistas and our understanding and appreciation of some of the commonalities and differences between normal glial cell differentiation and what goes awry in tumorigenesis. So very exciting to have you here and hear about your most recent work, Stacey. Welcome.
All right, I'm very happy to be here, and thank you for the opportunity to share my work with you all here today. So over the course of my studies, as Dr. Ph.D. mentioned today, I've studied both the development of neurons and of glial cells. And so I've studied the, oh, it's not on? Okay, just a second. Everybody can hear me now? All right. All right, so I started my journey into the development of the spinal cord in Dr. Jane Johnson's laboratory, where I studied the specification of dorsal interneuron populations. And what I found was that the transcription factor PTF1A was important for the specification of dorsal interneuron populations, specifically the DI4 population, and for the inhibition of the DI5 population. And later, during the second wave of neurogenesis, PTF1A is important for the specification of the DIA population, which ultimately gives rise to the dorsal horn GABAergic interneurons. And it's important also for the inhibition of the alternative fate, or the glutamatergic neurons. So when you knock out PTF1A, you completely lose dorsal horn GABAergic neurons. So for my first postdoc, I was still interested in neurogenesis, so I just moseyed on down to the ventral half of the spinal cord and did a postdoc in motor neuron development. And during that time in Soo-Kyung Lee's lab, what I studied was the function of a miR218, or a microRNA. And what I found was that the miR was expressed in motor neurons, and its expression was regulated by the motor neuron hexamer complex, which is LHX3 and islet1. And then later, they went on to show that it was important for the inhibition of the alternative or interneuron fate. But I was interested in studying something else. So, excuse me. We all know that the neurons are not the only cells of the CNS. There are the glial cells, and they, in fact, by far outnumber the neurons. But compared to neurons, we don't know nearly the same amount about their development and their function. So I went on to move to Ben Deneen's lab, where I studied glial development. And I'm sure I don't have to tell everyone here in this audience.
Welcome to Blue Jeans.
We're good?
To connect to the meeting using your remote control, please enter the meeting ID followed by pound or hash sign.
Okay. We're good? All right, so now we're in Ben Denin's lab, and we're studying glia. And so we all know that glia have all these important functions, and where the oligodendrocytes are the myelinating cells of the CNS and involved in saltatory nerve conduction. And astrocytes have many essential functions, including modulation of synapse formation and maintenance of the blood-brain barrier. So they have all these important functions, but we don't fully understand how they form during CNS development. So during CNS development, cell-fate determination in the developing spinal cords occurs in a spatial and a temporal manner. And in very broad terms, the space and the space dictates the patterning and the specification of neuronal populations beginning around E9.5 in the mouse spinal cord. Around E11.5, neurogenesis stops, gliogenesis begins, and that's called the gliogenic switch. So the processes that control the initiation of gliogenesis are really well defined, but the processes that govern the initiation of gliogenesis are not as well understood. But we do know that one of the important steps in the initiation of gliogenesis is the expression of the transcription factor NF1A. So NF1A is both necessary and sufficient for the initiation of gliogenesis and important for the continued inhibition of neurogenesis. So why do we care about the mechanisms that control glial cell fate? So we know that alterations in glial cell number or glial cell function can be found in a number of neural pathologies, including MS, a number of neurological diseases like Parkinson's, Alzheimer's, and ALS. But we also know that alteration in glial pathways have been linked to a number of oncogenic effects in brain cancers, including glioma. And I'm particularly interested in that because they have such devastating consequences for both children and adults. So pediatric gliomas are the most common CNS tumor in both children and in teens. And high-grade pediatric gliomas constitute about 11% of pediatric brain cancers and contribute to about 44% of the pediatric brain cancer deaths. So this really underscores the need for us to uncover effective treatment options. In adults, malignant gliomas have some of the worst 5-year progression-free survival rates, with GBM, or glioblastoma multiforme, having the worst prognosis. And even with our current treatment strategies of resection, ooh, what happened here? We're good? Okay. All right. Okay. With resection, radiotherapy, and chemotherapy, the median survival rate is only about 15 months. And I think one of the really striking things is that this median survival rate really hasn't changed in 60 years. So if we think about how we can tackle this problem, we really need to understand the basic biology behind glioma and behind gliogenesis itself. So some of the major biological processes that are involved in tumorigenesis are concerned with dysregulations of genes and pathways that provide the cell with a selective growth advantage. And we can broadly categorize these processes into 3 categories. And that's genome maintenance, cell cycle, and cell fate. So if we take genome maintenance, for example, this could involve mutations in genes like p53 that give the cell, allow the cell to escape apoptotic checkpoints. Cell cycle has a direct link to selective growth advantage, and this often have proliferation or vascularization components. And then cell fate processes have traditionally been viewed as having cell division or cell differentiation components. And these genes are often dysregulated rather than mutated. And that leads to an imbalance with it for the decision between cell division or cell differentiation. So most of the research into glioma have really focused on these two pathways, and that really has allowed the field to advance, particularly in the context of our understanding in the genomic landscape of glioma, but that's really not the whole picture. And that's really speaking to the fact that we've not really focused on how cell fate pathways affect glioma formation and progression. We know that the majority of cell fate decisions are made during development. So if we think about how these cell choices are made during tumorigenesis from a developmental perspective, then perhaps we can gain insights into how these decisions go wrong during the tumorigenic state and how they can be manipulated for effective treatment strategies. So another way we can think about this is that neural precursor cells give rise to either neurons or glia, and this process is in part controlled by the expression of transcription factors. For example, NF1A is involved in the generation of glial precursors and then later for the maturation of astrocytes. And not only do these glial determining factors have a role in specifying fate, but they've also been linked to glioma formation. So for example, Olig2 and NF1A have been demonstrated as having a role in glioma formation, indicating that there are common transcriptional requirements for glial development and for glioma formation. But although these studies have established that there are common transcriptional requirements for glioma formation, that whether their role in specifying cell fate can similarly influence the constituency within a tumor is largely undefined. And this is really an important unanswered question, because glioma is composed of different subtypes, like astrocytomas and oligodendrogliomas. They have vastly different clinical outcomes. So if we can correlate how the formation of these different subtypes is linked to the ability of these glial determinants to specify different glial sublineages, then we can perhaps make different treatment strategies for this deadly disease. So developmental paradigms are often reutilized during a malignancy, but how these pathways are reutilized is not really well understood. And that's particularly true for glioma, because we don't understand how the developmental pathways themselves are regulated. So what I'm really trying to say is that if we can understand how a glial cell is made, then we can better understand how a glioma is formed. So the approaches that I use to address these questions is understanding how the transcriptional regulatory mechanisms intersect with chromatin architecture during development, and how these processes contribute to glioma formation. And I used a variety of techniques, including molecular and biochemical approaches, and the model systems that I'm going to tell you about today are mouse genetics, the chicken ovo electroporation assay, which is a rapid and cost-effective way to test gene function, and then also the piggyback in utero electroporation model of malignant glioma, and that's a model that we developed in the lab, and I'll tell you more detail about this later in the talk. So today I'm going to go into detail about two different aspects of NF1A biology: what's upstream of NF1A regulation, so basically what regulates NF1A expression, and then also what's downstream of NF1A, so what does NF1A do in glial lineage. So let's first look at what NF1A does. So when I first joined Ben's lab, I started working on an ongoing project about NF1A in astrocyte lineage. And so I'm just briefly going to summarize what we found there. So the transcription factor SOX9 initiates the expression of NF1A, and that induces gliogenesis. After induction, SOX9 and NF1A form a complex, and that induces a subset of genes involved in the maturation of astrocyte fate. So after these studies, we were able to put NF1A in several stages of astrocyte lineage progression. But what I was more interested in is, what does NF1A do in the oligodendrocyte lineage? So oligodendrocytes have a very well-established lineage progression with well-characterized markers to mark those different stages. So the question was, where can I put NF1A in this already established network? So the first thing I did was to test NF1A expression in a temporal manner. So the expression of NF1A in the developing spinal cord at E12.5 early on in specification, I found that NF1A was co-expressed with oligodendrocyte precursor, or OPC, markers, PDGFR alpha, SOX10, and OLIG2. And then later on, as the cells mature, NF1A expression is maintained in the oligodendrocyte precursors as marked by SOX10 and NKX2.2, but its expression is extinguished as the cells mature. So NF1A is expressed in oligodendrocyte precursors, but not in mature cells. So I could put NF1A early on in the lineage, but its expression is not in the mature cells. So is its downregulation required for these cells to mature? If it is, then if I block that expression, can I block it by overexpressing NF1A? So the system I used to test that is the chicken ovoelectroporation assay. So I'm going to briefly describe that so everybody knows how it works. So you take your developing chick egg, you make a hole in the top so you can see it, and then you inject your DNA of interest in the developing chick spinal cord. You put your electrodes on either side, and then your DNA goes to the positive side of the electrode. That leaves your other side as your positive control. So now I have my system, and I can overexpress NF1A, and now in the presence of NF1A, we can block MBP and PLP, two markers of mature oligodendrocytes. So NF1A is suppressing oligodendrocyte differentiation. But how is it doing it? What is the mechanism? So NF1A is a transcription factor. So we looked at the promoter regions of various oligodendrocyte genes, and I'm just going to show MBP for simplicity, but we found this in various oligodendrocyte genes. So we found that NF1A binding sites in the promoter region, and using CHIP, or chromatin immunoprecipitation assays from E12.5 spinal cords, that NF1A bound its binding site in the promoter region. And then using luciferase assays, we found that NF1A could inhibit luciferase reporter activity, indicating that NF1A could directly suppress oligodendrocyte differentiation. So we could put its expression early on, and it seems to be directly suppressing differentiation. But we knew that it could be doing it in relation to some of the existing network. But how was it doing it? So we sort of took a clue from what we already knew from astrocyte lineage. So in the astrocytes, as I mentioned, NF1A is working with a SOXY family member, SOX9, to promote the astrocyte lineage. So could it be that NF1A was working with other SOXY family members to influence the oligodendrocyte lineage? So the obvious choice here is SOX10. So SOX10 is absolutely required for the myelin gene expression. So the question is, does SOX10 and NF1A have a relationship in the myelin or oligolineage? So the first question was, do they interact? So I tested for the interaction in Alinoid cells, which is a cell line that has oligodendrocyte precursor characteristics, and found that indeed SOX10 and NF1A do interact in this cell line. And then we found that SOX10 and NF1A bound to sites within close proximity to each other in myelin genes, in this case shown MBP. And then in luciferase assays, we saw that shown here MBP luciferase, that SOX10 could activate luciferase activity as expected. And then in the presence of NF1A, NF1A could inhibit SOX10 activity. So NF1A is antagonizing SOX10 activation of myelin genes in vitro. So the running hypothesis was early on in oligodendrocyte precursor cells, NF1A and SOX10 interact to inhibit the early induction of myelin genes. NF1A is downregulated, and then SOX10 can now exert its function to induce myelin gene expressions later on in the lineage. So if this is the case, then can we see this happening in vivo? So if we overexpress SOX10, what happens to the myelin lineage? So we turn back to the chick assay, and we overexpress SOX10 in the developing chick, and we see the expected induction in myelin genes, in this case MBP. And when we co-electroporate NF1A, we see the NF1A can inhibit MBP induction. So NF1A and SOX10 have an inhibitory relationship where it blocks myelin gene induction. But we thought that maybe they could have a cross-inhibitory relationship. So we asked the reciprocal question. So we overexpressed NF1A, and since NF1A promotes the astrocyte lineage, we see the expected induction of GFAP in GLAS, two markers of astrocytes. And when we co-express NF1A and SOX10, we see that SOX10 can inhibit both GFAP and GLAS. So this is suggesting that SOX10 can inhibit NF1A and thus astrocytes. And this is also indicating that maybe SOX10 is functioning in early oligodendrocyte precursor cells to suppress NF1A activity and thereby the astrocyte lineage. So what is happening, what can we see if we knock down or knock out SOX10 in mice? So we would expect that if we knock out SOX10, then we would affect the astrocyte lineage. And we were lucky enough to get a collaboration with Michael Wegner, and he sent us SOX10 knockout mice. And we then assayed these mice for astrocytes markers, GS and Aldose in this case. And this is the lower quadrant of the spinal cord. And we found that in the absence of SOX10, we see an increase in astrocyte markers. And this could have been for a couple of reasons. We could have just seen an increase in marker expression, or it could be that the cells that would have expressed SOX10 or OPCs are now becoming astrocytes. So we took advantage of the LAG-Z reporter, and then we assayed for reporter astrocyte marker double positive cells. And what we found was that there in the knockout mice, there was an increase in the number of beta-gal GS and beta-gal Aldose double positive cells, indicating that in the absence of SOX10, there was a conversion of the OPCs to astrocytes. So what we found was that SOX10 and NF1A have a mutually antagonistic relationship where they cross-repress each other's activity to balance the astrocyte oligodendrocyte lineages. So since these lineages are linked to glioma subtype, I wanted to know whether this relationship was important for glioma tumorigenesis. And I had previously, our lab and others had previously shown that NF1A was required for tumorigenesis. And we knew that NF1A was highly expressed in astrocytomas and GBMs, but nominally expressed in oligodendrogliomas. So I wanted to ask whether NF1A's ability to promote the astrocyte fate was conserved in gliomagenesis. So if NF1A is highly expressed in an astrocytoma, but not so much expressed in oligodendroglioma, if we overexpress NF1A in this oligodendroglioma, can I force it to become an astrocytoma? Now, this seems like a straightforward question, but human oligodendroglioma cell lines are very hard to grow in culture. So we had to come up with a model system in which to test this. So the model system in which we developed is a mouse piggyback in utero electroporation system that allows us to combine in utero electroporation with piggyback technology, where we can drive the RAS oncogene with Astro-glial lineages into the developing cortex. And if we use the CAG promoter, we can generate a histologically verified high-grade glioma in just 2 weeks. And then if we use the MBP promoter to drive RAS, we can generate an oligodendroglioma in the same time frame. And what's important for this particular experiment is that we can recapitulate the human oligodendroglioma, where we see high levels of SOX10 and little to no levels of NF1A. So now I have my model, and I can ask the question, does NF1A convert the oligodendroglioma to an astrocytoma? And remarkably, it did. So the top panel is our oligodendroglioma model, where we see oligodendroglioma-like histology, low levels of GFAP and low levels of the endovascular marker CD31. And then when we overexpress NF1A in the oligodendroglioma model, we see a shift in the histology to more astrocytoma-like, high levels of GFAP and high levels of CD31. And these are all properties of high-grade glioma. So taken together, this is indicating that glial determinants can affect glioma subtype formation. Now, this was really great and exciting, but I made cancer worse, not cancer better, right? So that's not really the goal. The goal is to help people. So I needed to do the opposite experiment. I needed to take a high-grade astrocytoma, which expresses high levels of NF1A, and delete NF1A and see if I could shift it in the opposite direction. And again, this seemed straightforward, but I didn't have the tools in which to perform that experiment. I needed to make a flox mouse because NF1A straight knockout mice die at birth. So I had to spend some time generating the NF1A flox mice. And then also our piggyback IUE is a really great system. 2 weeks, you have a high-grade glioma. It's really great for generating biochemical material and for doing high throughput screens, but it's not very good for monitoring your tumor over time. So I needed a more temporally tractable model to monitor my tumor progression. And so we all know that RAS is rarely mutated in glioma, but other genes that are mutated tend to converge on the RAS pathway. But genes like P53, P10, and NF1 are commonly mutated and have been used in the past to generate mouse models of glioma by Luis Parra's group. So what we did was we generated guide RNAs to these 3 genes and electroporated them in with our traditional model, piggyback model, and that allowed us to generate tumors in just 6 to 8 weeks. So to compare these 2 models, we have our RAS model that generates high-grade glioma in 2 weeks, and the model we're calling triple CRISPR generates a similar tumor, histologically verified high-grade glioma in about 6 to 8 weeks. And here's some quantification showing that we do see downregulation of the 3 genes or deletion. And then our survival curves show that we get a median survival rate with a triple CRISPR of about 90 days. So now we have a model in which to test it. So I take my flox mice, introduce Cree into the triple CRISPR, and found that we got efficient deletion of NF1A. And then the first pass through, I tested survival and found that mice that lacked NF1A had a better survival, which suggested that they might have a less malignant tumor. And then when I sent off the tumors to the pathologist, we saw that the mice that had NF1A had the high-grade glioma, and then mice that lacked NF1A had a mixed oligoastrocytoma with a high oligo component, no necrosis, and no proliferating, no vascular proliferation. So this is a promising result, and there's still much to do to understand the molecular and cellular mechanisms behind this phenotype, and this is something I'll be pursuing in my own laboratory. But still, it's suggesting that these glial determinants can influence glioma subtype formation. Yes?
How advanced is
It's stage 2 to 3. Sorry, 2 to 3. Yeah. So that's something I'm going to propose to do tomorrow. So this was my first pass through, just to see what would happen. So I took them when they were already dying, and so this was end stage about P70-ish, and so I don't know the answer to your question yet because I haven't done that experiment, but I'm hoping to basically knock it out after the tumor has already started. So one of the things I want to do is to take the tumor and do an inducible CRE, and then see when is it that I can delete NF1A and still see a conversion in the phenotype. That's the ultimate experiment that I would like to do. And I've started those experiments, but I haven't given them the tamoxifen yet. Yes?
You are introduced with an mutation in encrypted cells.
Yes. No, so these are given, we do this piggyback model from birth base, so they get given the triple CRISPR model in E15.5 embryo. So they technically have these modifications from birth. So that's why I'm suggesting that I need to do an inducible CRE where I make the tumor, and then once the tumor is already established, try to delete NF1A so that it's more of a clinically relevant model. So that is a caveat of these experiments, yes. Yes. So not in just particular these mice in general, but in our model, these mice do get epilepsy. But that's just a general thing in glioma patients. But these mice, triple CRISPR, wild type triple CRISPR, you can detect seizure in these mice prior to visual seizure. So if you give them an EEG, you can actually see them several weeks before you get a visual seizure of the mice. Any other questions? Okay. So to summarize this first part, the results show that NF1A can suppress the Oligodendrocyte lineage and promote the Astrocyte lineage both during development and during Gliomagenesis. And this really is reflecting a more nuanced role for glial determinants in oncogenesis, where the mechanisms that control sublineage diversification can similarly control glioma subtype specification, and rather than just general differentiation. And this is important because it has implications for how we treat the disease, because if we can manipulate the cell fates in a tumor to become either more differentiated or perhaps to convert the phenotype to be less malignant, that could potentially be a treatment strategy for glioma. Okay, so if we switch gears a little bit, NF1A has an important role during development and for gliomagenesis, so I wanted to understand more about how NF1A is regulated itself. So to understand that, I undertook a screen in the developing chick spinal cord to find regions that could recapitulate NF1A expression. So I aligned the chick and mouse genome and find regions of high homology, and then went on to assay those regions. So the first pass through this screen, I identified the E123 enhancer that recapitulates NF1A expression when fused to EGFP reporter. So when you electroporate that into the developing chick spinal cord, it drives expression to the ventricular zone, very similar to NF1A in the ventricular zone. And through a series of experiments that I'm not going to show today, we identified SOX9 as regulating the E123 enhancer. So when you overexpress SOX9 in the presence of the reporter, you can drive ectopic expression of the reporter. And when you mutate the SOX9 binding site within the context of E123, you lose reporter activity, suggesting that SOX9 controls activation of the E123 enhancer. So SOX9 activates NF1A expression through E123. But we knew from our genetic studies that SOX9 was not fully responsible for NF1A expression. That suggested that there were other factors also regulating NF1A. So to find out what those were, I went back to our screen and did another pass. And I found two additional enhancers that were modulating NF1A expression, and that's E161 and E96. So here's E123, which I just showed, and then E161, when fused to the EGFP reporter, also drove expression to the ventricular zone, similar to E123. And then the E96 enhancer drove expression of the reporter to the motor neuron area, similar to motor neuron expression of NF1A. And then here's the SOX9 that I just showed. And then through another series of experiments, we narrowed down that brain2 was regulating the E161 enhancer, but not the E161 enhancer, and that it was modulated through that brain2 binding site. And then I found that the motor neuron hexamer complex, LHX3 and islet, was regulating the E96 enhancer, but not the E161 or the brain2. E161 or E123 enhancer. And that this hexamer complex was required for modulating E96 enhancer activity. So altogether, what this data is suggesting is that there's lineage-specific enhancer activity regulating NF1A through distinct mechanisms. So just to summarize, E161 is regulated through brain2, E123 is regulated through SOX9, and E96 is regulated through islet LHX3. And SOX9 and BRAIN2 are co-expressed in the ventricular zone during the gliogenic switch, and islet is expressed in the motor neuron area. And given this expression pattern, and given how far apart these factors are and enhancers are in space, I wanted to ask two questions. One is, are these transcription factors genetically required for NF1A expression? And then since they're so far apart, how are they regulating NF1A in 3D space? So what is the 3D architecture of the NF1A locus? Yes? Oh, sorry. 100 and... So this is the dorsal interneuron populations. It's expressed in the DI3 and the DI5. And then this is the motor pool. So let's first look at the islet 1 knockout. And I found that in the islet 1 knockout, there was a significant decrease in the number of NF1A motor neurons, suggesting that islet 1 is important for NF1A expression in motor neurons. And then looking at SOX9 knockout mice, we had previously shown that in the absence of SOX9, NF1A was delayed, not lost, and that's shown here. Early at 11.5, there's no NF1A, but it starts to come back on at 12. And we see that same phenotype when we knock out BRAIN2. So we thought maybe if we knock out BRAIN2 and SOX9, they both might be required for NF1A. And so we do the double knockout mice, we see that we do not get induction of NF1A, suggesting that both SOX9 and BRAIN2 are required for NF1A expression. So given that these two enhancers, E161 and E123, are so far apart, I proposed this model where they're brought into close proximity to each other by this double loop structure. So if this double loop structure is occurring, then I should be able to show 4 different things. So 2 of them are that the SOX9 and BRAIN2 should form a complex, and the other is that this complex should occupy the enhancer regions and the promoter. So using co-IP assays from E12.5 spinal cord, I found that the BRAIN2 and SOX9 are forming a complex. And then using chip assays, I found that SOX9 and BRAIN2 complex are occupying the E123, E161, and the promoter regions. So then using CRISPR in the chick assay, I modified the binding sites for SOX9 and BRAIN2 in the context of the enhancer, and then electroporated the guide RNAs and found that when we modify the enhancer regions, we lose NF1A expression in the electroporated side. So this suggests that the integrity of the enhancers is important for NF1A expression. So then this gave me some confidence to go on and test for the actual presence of the chromatin loop structures. So the way you test for the chromatin structure is the chromatin conformation capture assay, or 3C. And the way this assay works is that you cross-link your DNA, you fragment it, and then you perform a proximity ligation, such that anything that's in close proximity to each other during the cross-linking is now fused together. You then perform a qPCR, and the frequency of your ligation product is then directly correlated to the long-range chromatin interaction. So then, using that assay, I wanted to test for the presence of interactions between the E123, the promoter region, and the E161. And then I suspected that there might be another chromatin interaction between the E96 and the promoter. So these are the 2 interactions I wanted to test. So then, to quickly orient everyone on how these assays typically look, you, in general, pick one region as your anchor point and then assay the surrounding locus. And this is then any peaks that you see represent a chromatin interaction. So in this case, I used the E123 as the anchor and then assayed the surrounding locus. And I found that when there's a long-range chromatin interaction between E123, the promoter, and the E161, suggesting that there was a gliogenic loop. And these are from spinal cord samples. And then I also tested for the presence of a motor neuron loop using in this case, the promoter as the anchor, and found that there was a long-range interaction with the E96, suggesting that there is a motor neuron loop. Now these are ligase-dependent reactions, so I did a ligase control and found that there was no long-range interactions in either case without ligase. And then to see if these were CNS specific interactions, I tested the lung. Now the lung also expresses NF1A, and I found that neither a gliogenic nor a motor neuron loop formed with any of the enhancers in the lung, suggesting that these loops were specific to the CNS. Now I was interested in trying to understand more about the gliogenic loop. So the question I wanted to know was, are SOX9 and BRAIN2 required for formation of this loop? Now this seemed like, okay, you should just do knockout mice, but the mating scheme for this would have required 729 mice at one time. So that wasn't going to happen. So what I had to come up with a workaround, and what I came up with was to use the HB9 GFP reporter mouse. Now this mouse is a beautiful mouse if you ever get to see it. It labels the motor neuron population. The motor neurons do not express HB9 or SOX9, so I manually dissected out the GFP and the ventricular zone. So the ventricular zone served as my positives, SOX9 and BRAIN2, and the GFP positive cells served as my knockout mice, so they do not have SOX9 or BRAIN2. So in this way, I could assay whether SOX9 and BRAIN2 are required for looping. So what I found was in both GFP and positive and negative mice, or cells, there were long-range chromatin interactions between the E123 enhancer, the promoter, and E161. So that indicates that looping formation is independent of SOX9 and BRAIN2. Now this is not completely surprising because these looping structures can form prior to induction of important developmental genes so that they can respond to developmental stimuli in a rapid manner. So to determine if this is the case for NF1A, I tested 2 additional time points, one prior to induction of gliogenesis and one well after. So I tested E10.5 prior to gliogenesis and E16.5 after, and found that at E10.5 there was presence of both long-range interactions between E161, E123, and the promoter, and that this architecture was persistent at E16.5. So this suggests that this gliogenic loop is a preformed chromatin architecture. So how is it that NF1A is induced then? So one idea is that it's the binding of the transcription factors that's inducing it to induce expression. So I tested whether SOX9 and BRAIN2 actually bound to the enhancers at E10.5 and found that they did not. So the white bars are the 10.5, and SOX9 and BRAIN2 did not bind to the E123, E161, or the promoter. And at 12.5, they were now binding to the enhancer regions and to the promoter, indicating that it's the precise timing of SOX9 and BRAIN2 that's important for expression of NF1A. So, of course, we want to know how important these gliogenic loops are to gliomagenesis. So one of the first questions we asked is, are they correlated in human gliomas? So we took data from the TCGA database and correlated NF1A with SOX9 or with BRAIN2 and found that they were indeed highly correlated. So then I generated tumors using the piggyback model and tested for the presence of the chromatin looping architectures and found that there were long-range chromatin interactions between the enhancers and the promoter, and it's very similar to the loop that we saw during developmental gliogenesis. So what happens when we manipulate these chromatin architectures during tumorigenesis. So this time I manipulated the enhancers in the context of the mouse using CRISPR. So I deleted SOX9 and brain-2 binding sites using CRISPR, co-electroporated them using the piggyback RAS model, and then assayed for long-range interaction, tumor growth, and NF1A expression. So first, I used the 3C assay and found that when I manipulated the enhancer regions, there was compromised looping formation. And this is indicating that the integrity of the enhancers is important for chromatin architecture. So, but what happens in vivo? What really happens to the tumor? And what we found was that there was smaller tumors, there was decreased NF1A, and there was decreased proliferation as marked by Ki67. And then we monitored tumor growth by bioluminescent imaging and found that there was decreased signal, indicating that the tumors were indeed smaller. So overall, what we found was that the chromatin architecture of the NF1A locus appears to be a key regulator of gliogenesis and glioma tumorigenesis. Yes?
When you check the pathology of the tumor, the oligodendroglioma, after the loss of NF1A,
So there's still some expression of NF1A, and there's still they're still high-grade glioma, but they didn't tell us whether they just called it high-grade glioma. They didn't give us a subcategory. But technically, the mixed Oligoastrocytoma is also a high-grade glioma. Yes. Yes, that is true. But this one, they didn't categorize it in that detail. So I don't know the answer to your question. Right. So to summarize the second part, what we found was that their glial transcription factors regulate sublineage diversification both during development and during gliomagenesis. There are lineage-specific expression of NF1A during spinal cord through distinct chromatin looping mechanisms, and this chromatin loop is conserved during gliogenesis and gliomagenesis, and the integrity of the chromatin architecture is important for tumorigenesis. And so in the last few minutes, I'd like to briefly share my plans for my future laboratory, and I'm going to be using the tools and techniques that I've developed over the last several years to address questions that pertain to the larger question of how transcriptional mechanisms in chromatin architecture interact and function to regulate cell fate and development and disease. And the more specific questions that I would like to address are what are the mechanisms of chromatin looping in the CNS? And as I mentioned, SOX9 and brain2 are part of this glial looping structure, but they're not required for formation of the loop. So the question I want to address are what are the mechanisms of this chromatin loop structure? So how is this loop forming? And then can I exploit this SOX9 brain2 complex to identify other loop structures, other genes that are involved in gliogenesis? So basically, can I find other genes that are not NF1A involved in gliogenesis? And then I'd like to understand how these glial determinants are influencing glioma cell fate. So basically follow up on that phenotype, how NF1A is converting glioma subtypes. And then, as I mentioned, there's this glial loop, and then there's the motor neuron loop. So how are these two loops different? Are they regulated in the same way? Do they form in the same way? And then also, what is the function of NF1A in motor neurons? So just to give a little bit more detail about each one, I have some preliminary data that suggests that the chromatin architecture protein MED12 is involved in formation of this loop structure. So I'd like to determine what the role is of MED12 in formation of this loop. And then more broadly, what is MED12 doing in the astrocyte lineage? And then what are these developmental mechanisms involved in glioma tumorigenesis? And then I performed several screens to begin to understand how the SOX9 brain to MED12 complex is involved in gliogenesis, and to begin to understand how other genes are involved in gliogenesis other than NF1A, so to begin to move away from NF1A. And then to use mass spec to begin to understand how these chromatin looping mechanisms are working in both development and in tumorigenesis. And then to understand how glial determinants are working in glioma subtype specification, I'd like to fully characterize this NF1A loss of function phenotype. Then to do, as I mentioned, these late stage or delete NF1A in a more preclinical type model. And then to begin to understand the molecular mechanisms of NF1A in gliomagenesis. And then lastly, to understand how the motor neuron loop and the gliogenic loop are different. Are they the same? How are the mechanisms of chromatin architecture functioning in a lineage-specific manner in the CNS? And then understand what the function of NF1A is in motor neuron development. And with that, I'd like to thank Ben. He's been very supportive throughout the whole career, especially when I decided to switch into the glia field. Everyone in the lab, especially Jeff and Wendy, who helped me to set up the piggyback in utero electroporation model. Carrie, who's our pathologist. Michael Wegner, who sent us the SOX10 mice. Sue Kyung Lee, who sent us the islet-1 knockout mice. Chad, who is the bioinformatician, and our funding sources. Thank you very much. Yeah, so I think in that case, we can, the way that we did the deletion, it was not a point mutation. We debated a lot about how to do it, but in the end, we ended up deleting about 200 base pairs. So I think that it's actually the integrity of the enhancer that's important. So it's not necessarily about the binding site of SOX9 or SOX10. It's about the integrity of the enhancer, the spacing of the enhancer. So in other systems, when you delete the enhancer that are these preformed loops, you can lose the looping if you delete the spacing of the enhancer. Right. So I suspect that since they're not required, that may not affect formation of the loop. But I have not done that experiment. Yes. So it's involved in many cancers: prostate cancer, breast cancer, melanoma. Different mutations cause different things. It's been implicated in glioma. There's IDH mutant, for example. How should I say? When you have an IDH mutation, it causes one enhancer to now be available to affect PDGFR alpha, which normally it would not be available to do that. So that is one of the pathologies involved. So you can see that in other cancers as well. So chromatin architecture modifications have been identified in other cancers, yes.
Is it generally considered to be sufficient to trigger the cancer because the immune structure?
In some cancers, yes. In others, they don't know what the actual mechanism is, but they have seen chromatin architecture effects. There's actually mutations in chromatin architecture proteins like CTCF, cohesin, and those are causative.
So, great talk. I want to go back to the beginning of your talk where you talked about the role of NF1A in oligodendrocyte development, and that it's present in the OPC and seems to stall them from undergoing myelination. And I wanted to know whether you looked at plaques in MS where there are OPCs, but they fail to remyelinate. Is NF1A dysregulated in those plaques?
So,
Targets that could be knocked down and get remyelinated.
so we had a paper in 13 or 14 where we overexpressed NF1A. So they are expressed in OPCs in MS tissue. We had stainings for that. We overexpressed NF1A in lesions. So we did the IPS. We did. Let me see if I have this. I might actually have it here to show you. Here it is. Okay. So this is HIE. So here's NF1A with PLP in human the HIE patients, and here's the MS plaques. So you see it's in OPCs in the MS lesions. But it's, when we did the lyso model, where you inject the lyso and it causes a demyelination, we see that NF1A is expressed in OPCs, but it's not expressed in the mature cells. And then what we did after that is we overexpressed NF1A using virus in this model system, and it actually blocked repair. So here's the virus, and here's the control, and it blocked repair of the lesion. So it can even block in demyelinating lesions.
Just a quick question. So with regards to, you just hinted at this, I think, so with regards to the proteins that are actually mediated in terms of movement, looks like you have one candidate, NED12. Have you looked at CTCF proteins and YY1 proteins that have been indicated in one that comes to roots and other tissue types?
So CTCF has not been implicated in promoter binding. So, and the other thing is that there's been reports in the brain that it's not expressed in glia. It's expressed in neurons. So I didn't test it for that reason. The other reason that I picked NED12 is it's been shown to bind to both SOX9 and to brain2. So, sorry, not brain2, sorry, SOX9 and SOX10, protein-protein interaction. And so I felt that it was the most likely candidate, and it worked out that it actually chips, actually co-IPs, and is expressed during the correct time frame. But I have not formally tested CTCF. And the other reason, too, was that there's no CTCF binding sites contained within the NF1A genomic region that I was looking at. So I didn't think that it was a good candidate for me to test because there were no sites. Usually there's a CTCF site for your TAD boundary, right, for your topological boundary. But there wasn't one anywhere near the NF1A 120 kb. So I didn't think that it was the best candidate. Yes.
Last question.
So you showed that blocking NF1A is used to oligodendrocyte. I've never seen in the Neuroscience the from the root structure has been lost.
Could you say that part again? Have I ever seen what?
Have you seen the from the root structure in lost over the structure to lead to the
So no one has tested any, to my knowledge, chromatin looping past the neural precursor stage. It's actually what I'm going to propose tomorrow. And that's one of the things I'm interested in looking at. This is the first report of any chromatin structure in neurons. I mean, in CNS, other than some NECP2 stuff.
Please join me in thanking Dr. Glasgow.